Thank you. It's a great pleasure to be here. BioIT World is one of my favorite conferences because every person in this room matters and you can make a big difference. So for me, it will be a great pleasure today to talk a little bit about where generative AI is today in terms of the capabilities in drug discovery. And I'm going to talk about my favorite topic, which is aging research. Because as James mentioned already, people matter. And my life's goal is to make sure that people live longer. That's the only thing I care about. Actually, you cannot live longer without living healthier. So it's kind of the same thing. And currently, my problem in life is that there are so many ways to diagnose aging. You can look at yourself in the mirror or in your passport. But there are very, very few ways to intervene in aging. So there's nothing clinically proven. And we want to figure out how to change that with generative AI. So my question when I started Insilico was, can we develop dual-purpose therapeutics that can target aging and disease at the same time? Because the hypothesis is if the drug is powerful enough to work on aging, it should work on many, many diseases. So that's the key. And I encourage many of you here in the audience, especially the people who are starting their career, to look into this hypothesis deeper and to align your careers with aging-focused drug discovery. This is a little bit about the company. So our company is in the business now for 11 years. Well, it's our 11th year. We originally started in Baltimore at the ETC at Hopkins, and then we kind of scaled. So now we're local to Boston, but we are truly global. And we utilize the power of AI and also the power of the infrastructure around the world to very quickly discover and develop molecules and push them into clinic and through clinical trials. So the company can be explained in just one slide. That's the slide. It took me a while to kind of figure it out. We do AI. I call it software for science. So you can buy most of what we do as commercial tools, or much of what we do is also openly available, open source, some of the cool models, frontier models. And we span biology, chemistry, medicine, and also all things science. Pandomics is a very popular tool that many of you use for target discovery. It has many, many different target philosophies in there. You can start from scratch or put your own data. Chemistry 42 is a tool that allows you to design small molecules with the desired properties from scratch or from a ligand. There is generative biologics that allows you to generate novel antibodies, nanobodies, peptides. We also do clinical trial outcome prediction, and we also, something that I'm very passionate about, we also do a lot of agrochemistry, petrochemistry, and CO2 capture. So materials designed for sustainability. And we utilize our entire software suite to actually develop our own drugs. And every drug discovery exercise that we have is an experiment to see if something in a new feature in the pipeline, in the drug discovery pipeline on the platform side, works or not. And that's the idea. So actually bet our own money to see if our software works. For many of you who I encountered in the exhibition hall, by the way, beautiful exhibition hall this year, highly encourage everybody to visit. I see that you're basically trying to empower pharmaceutical companies to test your software. We actually bet a lot of money to actually see if the molecule works or not. And sometimes the level of novelty of the target that we go after is very high. So TNIC, for example, has never been in a clinic, and there were no selective chemical molecules against it, so we had to go from scratch. But when you're going too novel, usually you will have to go phase 2 because otherwise pharmaceutical companies don't believe in a hypothesis. And sometimes you need to find moderately novel targets that are commercially tractable and are maybe not, may not have clinical POC, but there are some front runners in front of you. So you can sell those molecules very quickly to pharmaceutical companies if your molecule is truly best in class. And that's what we've done. So we licensed out USP1 to Exelixis. It was an $80 million cash upfront deal, pretty substantial downstream. And we sold a few others just recently. So if you follow us, that's kind of the idea, to pre-cook the cookies and sell them to the cookie makers while empowering and testing your own cookie making software, and then making the software available to the community. So if you look at the pipeline, this is how it looks like. Our most advanced one, from 0 to phase 2A complete, is in idiopathic pulmonary fibrosis. The reason why I decided to go after IPF is, well, first of all, the target scored very highly in lung fibrotic diseases, but also it's a disease of old age, so average age of onset is 65. And we also did the biomarker study using Olink proteomics to see if, well, first of all, if our antifibrotic hypothesis is confirmed, but also we're measuring aging clocks. The data is unpublished. Hopefully later this year we'll be able to present that. And now we've managed to take a bunch of programs into human clinical trials from scratch very quickly by utilizing generative AI. And the greatest challenge in drug discovery is actually experimental validation. So unfortunately, unlike text, music, voice, video, we as humans cannot really validate the output of generative systems in biology or chemistry. So you need to perform the experiment. That is why very often on the software side, it takes me very short period of time to develop a software piece, but then it takes you years to validate. And now we try to, so here you have some internal Insilico benchmarks. This comes from my annual presentation to my staff, saying that, look, right now with AI coding, AI startups can develop a product with less than 30 days. So 30 days to product. And then you basically launch and test, right? Many of them, they lack the brand or reach. That's why they are a little bit slow, but a lot of products are coming. And for us, currently the time to prototype is 4 months, time to product. Usually we try to go after we validate several models experimentally, we put it on the market. But still, over a year. So that's a lot of time. So we try to accelerate that, but we also try to accelerate drug discovery. And drug discovery, I think that currently we have already accelerated to the limit. So unless we see some regulatory changes, it will be very difficult to do it faster. And now we also, so another problem I have on this side is, on the AI side, is that the drug discovery team needs to work slower, but you have to push the AI team. And the AI team can be pushed only by closely integrating with the drug discovery team. So we decided to actually enable everybody in the organization with generative AI. And now part of the organization is run by generative AI, by an LLM, because LLMs can reason extremely well, not only in terms of therapeutic programs, but also in terms of producing novel software. And one of the tools that we very rapidly developed and put on the market, you can actually subscribe to it for, I think, like $17 a month. If trial is free. So we decided to see, okay, what is the key bottleneck that every scientist faces, and that is writing scientific papers. So we decided to open part of what we have internally when we run the company using AI to the world. So now using Dora, it's our tool for automated paper writing. You can deploy armies of agents that will perform basic bioinformatics research and then write a paper for you from scratch with original references. Pretty cool stuff, but we use the same idea in-house to even run the robotics lab. So you just treat each piece of hardware as an agent with an API that you can instruct, and it will perform certain tasks. So that's an extension of Dora. So have a look at it. This is how the company is organized. So that comes from my 2023 December presentation to my staff. So we have an Insilico commander, an LLM that can reason and organize and orchestrate other models. But in-house, we have over 800 different models that are validated for specific tasks. And very often this task is physics, so LLM cannot do that, or some specific biological model, but it has an API that LLM can access and orchestrate. And currently we also have a lot of feedback from the community of users where we are constantly improving the software tools and also improving our own drug discovery programs. This is the most important slide of the day because the idea was to tell you what's kind of where we are with drug discovery. So in 2019, we raised our first significant round to try to go from algorithm to drugs. And then we started timing. So looking at how long would it take us to get to preclinical candidate. Our preclinical candidate or developmental candidates is the same thing. So it's defined by lots of efficacy data, usually at least 2 animal models with efficacy, and at least 2 species with 28-day non-GLP tox. So far we have not failed in GLP toxicity studies a single time. So 100% success in IND enabling. And so far we have not failed in single phase 1. And now we have phase 2 complete also with safety, with good safety and tolerability. So that shows you the typical kind of time that it takes us to go to developmental candidate to this stage where you have a PhD defense with safety and efficacy data and defend your molecule. You also need to show that you're best in class if you're fast following something. So far it takes us approximately like 13 months to go from 0 to developmental candidate. I also must say that part of this reason is China. So we also have boots on the ground in Shanghai and Suzhou and other regions working within CROs to quickly produce the data and ensure that the FTEs are doing what they need to do and you get the best FTEs. And we have a network of over 40 CROs that very often perform experiments in parallel. And sometimes redundant experiments. So if one lab fails, another one succeeds, we have to redo, and that adds a few more months to the timeline. But those are the realistic timelines. And now we have reached a stage where we don't want to accelerate on too many developmental candidates unless I sell a few. So now we have to license out. And I'll show you a few. So this was the most important slide today. But I'll show you a few other examples where AI can perform really well for drug discovery. So our journey started in 2018 with experimental validation, and many of you have read this paper, I hope. So one of the people here approached me and said, look, I actually chose the career in bio-IT because I saw this paper. It was the best comment I've received this week. In this paper in 2018, we performed the experiments. It took almost a year to publish. We used generative tensorial reinforcement learning that we've developed with Alana Spurogudzik before at Harvard, now at University of Toronto, and we used to be fierce rivals from 2016 to 2018. Now we've partnered. And this paper got pretty popular. And in this paper, we showed that if you pull a target out of the hat, in that case, it was DDR1 kinase pulled by WuXi AppTec. We wanted to ensure that we go after a kinase because we had only one shot at it. And we had to sacrifice novelty for synthetic accessibility. But very quickly, in 21 days, we designed 6 molecules. 4 of them worked in binding studies and in binding affinity studies. 2 went to microsomal metabolic stability studies, also in vitro validation for efficacy. And 1 reached the mouse in 46 days. So that paper was criticized by the kind of old school academic community. You know, Mark Murko used to be my hero, Relay Therapeutics now. And they said that, oh, it's not a super novel target, maybe not super novel molecule, but it was the first experimental validation of this kind. And we had to prioritize at that time synthetic accessibility, because usually you don't have a lot of money to synthesize. We synthesized only 6 molecules. We responded. And recently, there was a paper by Tom Blundell's lab. I didn't know about this paper, but that's very, very cool, important paper if you're getting into this field from scratch and learning about it. So they basically went and did the literature review for all the generative experiments that were published with experimental validation. And table 4 there, it's also available on GitHub, so they're constantly updating it. It shows you the kind of input, output, design task, target, hit rate, outcome, and publication year. And you can see which generative models, so in our case it's generative reinforcement learning, so it's goal-oriented learning. Much of what we have done here was published and validated. So I was very happy to see that the table. And also now they have many more models. So if you're actually interested in picking the right model, you might want to go to their GitHub and see. Can we generate compounds for proteins without the crystal structure? It's a good question that everybody is asking. So in 2021, we did this experiment. We decided to use our target discovery tool to find a few promising targets that didn't have a crystal structure. It were quote-unquote dark targets. Used AlphaFold, the previous very old version of AlphaFold. Did some molecular dynamics, gave it to chemistry 42. In 2 rounds of synthesis, we got to 180 nanomolar molecules and validated in vitro and published in Chemical Science. So the entire experiment took 50 days. It's pretty cool. So 50 days from 0 to validation without a crystal structure. Of course, it's not like the developmental candidate path molecule, but it shows you the capabilities of generative AI today. Can we use AI to repurpose existing drugs? Here is one example of this kind of experiment where we don't do repurposing in-house, we do only novel, but for supporting others, sometimes we try. So I gave a talk just like this at one of the really cool conferences, ALS Consortium in 2022, where Merit Chutkovits and Jeffrey Rothstein were present. They had the Answer ALS Consortium with a lot of data from ALS patients. We took that data, combined it with our data, gave it to Pandomics, very quickly identified some novel targets and drugs to repurpose. And Jeffrey Rothstein recommended his former student, Zhavoronkov, who has the fly model for ALS. And we very quickly tested and published within 4 months a paper with predicted targets with validation using RNAi experiments. And in that paper, we showed 18 targets that worked extremely well out of the 26 predicted. And Bailu, very famous Chinese-American neuroscientist and a past dean of the Tsinghua Medical School, he has a company, co-founded a company called 4B Therapeutics. They took one of the targets and one of the repurposing candidates, and in 1 year, they enrolled 64 patients in ALS in IIT. It's an equivalent of phase 2 investigator-initiated study. And it just completed. I cannot talk about the results, but it looks like very promising. So the manuscript will start drafting soon. And it shows you that with generative AI, with repurposing, within 2 years, you can go from 0 to, 2.5 years, from 0 to phase 2 equivalent complete with POC. So that will be a really fun paper to read. And what you need to know about Insilico is that I'm very focused on aging, right? So I pretty much don't care about anything else because that's 100% guaranteed to kill you, right? So I'm not gonna, burn a Tesla if I'm not agreeing with somebody. It doesn't matter, right? What matters is how long can we push the human lifespan. So many products that we developed also can be used for aging research, and many targets can be repurposed for aging. Again, my big hypothesis is that if the drug is powerful enough to work on aging, it should work on many diseases. It's the path to blockbuster drugs. Currently, I think that GLP-1s are also anti-aging drugs, maybe can give you, half a year or a year in biological age advantage. But if you are, with a disease, it will give you even more. The problem currently is that there are no drugs that are clinically proven to work on aging, and we need to fix that. Now I have the same problem, so push hard. Next slide. Can we fix the clicker? Okay, So the idea for target discovery that we published several times is to find targets that overlap in aging and disease at the same time. So you construct models that basically have some representation of how aging works, and then look for most important targets that may be also implicated in diseases. So in 2022, in January, so the work was done in 2020, we published a paper how we can annotate all the targets that are implicated in aging and disease at the same time using the data available at that time. We took the data from longitudinal data from people with diseases and also healthy people, and identified targets that score very highly in diseases and also in hallmarks of aging. And I'll have to accelerate because I have a lot of slides. And in this paper, we did this kind of diagram where we structured and classified the targets by novelty, drugability, and also implication on certain pillars of aging. It's called hallmarks. So things like biological processes, like cellular senescence, inflammation, mitochondrial dysfunction. And many of those targets are very well known, so you can repurpose them for experiments, and some are very novel. So one of the moderately novel targets was TNIC, and we decided to go after this target. It's very new. Some people were toying around with it in cancer before, but nothing went to the clinic, and not that many papers. And we published a paper recently with Colin Ewald at the University of Zurich. Now he is the director of aging research at Novartis, showing that this target can be positioned in cancer, metabolism, age-related diseases. How can we validate something like this, right? So there are many ways to do that. So you want to find many different angles that support your hypothesis for the target. And one of the areas that also helped us support the hypothesis for TNIC is life models. So life models is something that is in AI excites me the most. So that's what I work on the most. That's an extension of world models. So world models, if you are not familiar with world models, I highly recommend this paper by David Ha and Jürgen Schmidhuber, which explains that if you create, you don't need to have a perfect understanding of the environment in order to create the environment where the agent can perform useful work. And they did a POC in imaging with that. And now David Ha is head of Sankana AI in Japan, and Jürgen, of course, is a celebrity as he is. That idea has been expanded into many other papers, and you probably have seen Sora by OpenAI, right? So it doesn't have a perfect understanding of the world, but it can create a 1-minute representation or 1-minute video from a prompt like this or like this. It's a very old stuff, like 1.5 years old. But it's still pretty impressive. So we thought, okay, well, why don't we do the same in biology? Build models that understand the life from cradle to the grave. And we published a bunch of papers on that, just on a hypothesis, and then started publishing experimental work. So how you can utilize longitudinal data coming from different sources, publicly available, previously incompatible, that you feed into deep neural networks and train generative models that understand life itself and understand the intimate relationships between, for example, methylation and transcriptomics, so central dogma. But we realized that one universal feature that can unify those data types is age, because everybody in this room has age. Not everybody has cancer, somebody probably does, but everybody has age, and that is a feature that actually defines when you die. So it's a very cool way to test the systems. So if your generative system cannot predict age without knowing your age, well, it probably wouldn't know anything else, right? So we basically use it as also for validation for reinforcement learning. And we published a bunch of patents on this. I'll skip a few things. And one model that impressed me a lot in 2022, I was presenting at the GRC meeting on systems aging in 2022. And a few months before, Gatto came out by Nando De Freitas at DeepMind. beautiful model that allows you to achieve full multimodality on multiple tasks. And we thought, wow, this is what we want to do with live models. And we decided that we're going to develop a model, transformer diffusion-based. We call it Precious because it's one model to rule them all, that can perform multiple tasks, that can predict age, generate biological data with age as a generation condition, annotate the results of omics experiments, etc. And now we have published multiple Precious series. Our Precious 1, it's published, you can check it out. It was used for target ID and age prediction. Precious 2 was used mostly for synthetic data generation and creating digital twins into the future. And Precious 3 is an open source model. It's a multimodal transformer that is trained on methylation, transcriptomics, proteomics, and text. It's instructional, and you can use it for many different tasks. We opened it to the community without putting it in the peer-reviewed journal yet, just to see how people use it. So it's trained on many different data sources, of course, like Link, Geo, Wikibiobank, etc. And you can use it for drug discovery, for aging research, for, if you like to do herbal medicines, you can do herbal medicines with that. And you can very rapidly validate. So I'll skip a few. It's open access, so have a look and take it. You can play around with it. You can play around with it on GitHub, on Hugging Face. And this model also understands non-human data. So we basically put in mouse, monkey, and human. And if you show it dog expression data, it actually does zero shot from dog. So that is a pretty cool idea. So it's a multi-species system, already on the market for almost a year. So a lot of people use it using kind of different philosophies. And in order to validate those systems internally, we decided to build our own lab. We built it in Suzhou during COVID, so I had to bubble there. Can you play the video? Because it doesn't start for some strange reason. But if we fail to do that, you can find it online. It's an epic robotics system that you walk in. It's real footage, not animation. It recognizes your face. You've got dimmed windows. You can undim them, see the robots. And it has multiple workflows. So here is one. So you throw in tissues, cell, organoids. The system automatically picks it up, grinds it, microplates it, does quality control, passes it to another room. There I have an army of autonomous guided vehicles. They transport the samples around the lab. Part of the sample goes into the incubator, part into the imaging station. Do 4 or 5 types of imaging, Celigo, CX7s, etc. Then for every sample, we usually do 3 library preps, so methylation, transcriptomics, and whole genome. Then Illumina sequences this data, and all this data comes into our PandaOmics-like system that performs the magic. So based on your novelty criteria and assessment of the target criteria that you want to have in a perfect target, the system picks a bunch of targets, looks for those that also have repurposing tool compounds, picks them from the compound hotel, incubates the original samples with those molecules, and then evaluates the readout, again using imaging, so phenotypic and methylation transcriptional response. If AI did well on target ID, it gets rewarded. Otherwise, it gets punished. So it's complete. It's complete kind of reinforcement learning loop with a lab in between. And also, if a lot of compounds scored well, but a lot of targets didn't have the compounds, and they scored even better, we go deeper into the hypothesis using CRISPR and RNAi. And also other experiments. And now I'm actually automating this lab even further. It's a lot of fun. This is my new baby, our humanoid. So now we're training embodied AI models that allow humanoids to actually perform different laboratory tasks. So currently it's early days, but I'm pretty sure end of this year you would already be able to pipette and hopefully beginning of next year, we'll be able to hook up our DORA to the humanoid and completely automate the workflow, even for the equipment that is not meant for automation. So now I'm almost out of time, so I'll have to skip a few things. But now I just want to leave you with just a couple slides. So imagine that you found this novel anti-aging target that was confirmed using multiple experiments, and you need to somehow put it into the market, right? First, you need to fundraise. So you need to convince people that it actually works in a disease. And the entire history of how we did that for one of the targets is published in my most important paper to date, published last year approximately same time, in March 2024. It's a small molecule TNIC inhibitor, targets fibrosis in preclinical and clinical models. And in this paper, we describe this entire process. So how we identified the target, how we did the hallmarks of aging, how we did the synthesis of a novel compound. The molecule is disclosed, by the way, figure 2 if you're asking. And then performed many, many different experiments. Performed phase 0 in Australia in healthy volunteers because the target is new and we wanted to do microdosing. Then we did 2 phase 1s. And then we just completed phase 2A in China. And we just got a big paper accepted in May. You're likely to see the phase 2 results. The phase 2 results surprised us in a most dramatic way, because if you know IPF, usually whatever you do, you decline. So for us, at 12 weeks of dosing, we saw dose-dependent response, and at high dose, 98 milliliter increase in forced vital capacity. So that was pretty surprising, and that data is going to be published in a big journal soon. So in that particular study, we show that we identified the kind of aging pathway, that targeted it, purpose towards a disease, and then we also show massive repurposing potential in multiple other diseases. Of course, first we wanted to get it approved for IPF. You know, if we can get it approved. Currently, the problem with generative AI is that you can accelerate preclinical development by 2.5 years probably. But in the clinic, you have to move with the speed of traffic. You cannot like cut corners. So hopefully we'll see some really cool results end of this year, because more clinical trials are kind of starting and finishing. And if you are interested in an application of a robotics lab to the demonstration of geroprotective properties of this drug, read this paper. It just dropped in Aging and Disease, showing that our 055, now it's called rintosertib, has anti-aging properties, and how we did it, how we confirmed it. And if you're interested in kind of one more exercise, we just also published it in Nature Biotechnology in December. We found another target that is very implicated in aging. This is not a new target, so PHD1/2 is super well known, but we think it's very strongly implicated in aging, and we purposed it towards IBD, inflammatory bowel disease. And in this one paper, we show all the way to from 0 to IND enabling complete. And now we have also 2 phase 1s complete with very good safety. So here I'll have to end, and kind of shameless promotion of our non-profit volunteer-run conference. Every year we organize aging research and drug discovery event in Copenhagen. It's very cheap. It's 5 days, 180 speakers, and there we bring pharma and academia to discuss how to develop drugs for aging. But of course, with the diseases in mind. Thank you very much. Open to questions.